Hi, I'm Haseena from IIT Delhi, India. It's a pleasure to be a part of this symposium to introduce you to my research on catalytic reactions in complex environments to produce biorenewable energy and chemicals. Over here, I'm showing the typical processes like pyrolysis, gasification, and hydrolysis, which I call as technology A, that is used to extract chemicals from biomass. However, a major drawback of these processes is that it requires harsh reaction conditions. Herein, we introduce technology B, that is integrated bio and chemocatalysis, which combines fermentation or biocatalysis with chemical catalysis to produce chemicals and fuels in a sustainable fashion. In this technology, waste biomass is fermented using microorganisms in a bioreactor to produce platform chemicals. These molecules can further be purified and chemocatalytically upgraded into useful chemicals. We propose 6-amyl alpha pyrrone or 6-PP as a potential platform molecule which can be produced from the fermentation of sugarcane bagasse using Phycoderma atrovidae species. The graph over here shows the current 6-PP production status in our lab. So far, we are able to produce 742 milligrams per liter 6-PP by applying an extractive fermentation technique. The 6-PP thus produced can be further purified and chemocatalytically converted to a variety of chemicals as shown over here. We have explored ring opening and decarboxylation of 6-PP to produce non-2-ene-4-one, which is used as a flavoring agent. And this molecule can be hydrogenated to produce linear alkanes, which can be used as fuels or fuel additives. The reduction of 6-PP produces diol molecules that are important monomers in the polymer industries. Apart from this, these reactions, 6-PP can be catalytically hydrogenated to delta decalactone or DDL, which has potential to be a platform molecule by itself. This reaction in particular is very interesting. As you can see here that we get almost 100% conversion in just 10 minutes reaction time yielding 79% DDL. Isn't that amazing? And yes, we have used formic acid as an in situ source of hydrogen instead of hydrogen gas, which makes the reaction even more greener. To draw more details, we performed this reaction in different solvents and we observed maximum conversion in cyclohexane. In addition to this, a microkinetic model was developed to understand the correlation between the temperature and hydrogen production from formic acid. With increase in temperature, the hydrogen production increased steadily, and we saw that the 6-pp conversions correlated well with this trend. And the conversion attained a maxima at around 433 Kelvin, as shown here. All these results that I have discussed so far were done using commercially available 6-pp, that is pure 6-pp. However, towards integrating the processes or technology for bio-based DDL production, we started the reaction, the hydrogenation of fermentation-derived 6-pp. We used 6-pp that we obtained from the fermentation process. This time, however, we saw a decreased conversion, that is about 63% conversion. And more surprisingly, we did not observe any DDL formation. So we assumed that this could be probably due to the impurities present in the 6-pp feed that we have used. And also from the literature, we could see that biogenic impurities that comes from the fermentation media or like vitamins, proteins, and amino acids, which are basically added as a nitrogen source into the fermentation media or is produced as a result of the biocatalytic processes, potentially interacting with the catalyst surface and deactivating them. So we assumed that the observed deactivation in the 6-pp hydrogenation reaction could be due to the impurities present along with the 6-pp. In order to validate our assumptions, we performed some model impurity experiments. So by model impurity experiments, what I mean is that we introduced less than 100 ppm of model impurity into the same hydrogenation reaction which we had performed. And we saw an interesting trend where sulfur bearing amino acids show deactivating the catalyst significantly. And the cysteine molecule showed the highest deactivation among all of them, while alanine, which was a sulfur free amino acid, did not show any effect on the conversion. And more interestingly, the product yields were also significantly affected by the presence of sulfur bearing amino acid. And it seems that the hydrogenation pathway is completely shut off in presence of sulfur bearing amino acids. So, in order to understand the deactivation mechanism, we used density functional theory simulations. And over here, I'm showing you the comparison of binding energies of 6pp and amino acid molecules. And you can see that cysteine has a very strong affinity to bind on the PD111 surface. And this could be probably leading to a competition between the reactant molecule 6pp and the impurity cysteine to adsorb on the surface and causing deactivation. So, in order to understand the cysteine deactivation, we performed a detailed mechanistic study of cysteine on PD111 surface. And as you can see here, cysteine readily dissociate on PD111 surface and form atomic sulfur. This explains the strong catalyst deactivation that we observed in case of experiments. And also, we performed some molecular dynamic simulations to understand protein interaction with PD111 surface. We used BSA as a model protein, and we saw that protein molecule aligned with some of its cysteine dimer linkages to the PD111 surface. And this observation is also in correlation with the experimental results that we saw that BSA caused moderate deactivation. Probably that is due to the large size, which prevents the molecule from diffusing into the catalytic active sites to cause deactivation. So, to summarize my research, we explored the catalytic hydrogenation of platform molecule 6-amyl alpha pyrrone, and we saw interesting solvent effects. We also observed significant catalyst deactivation from sulfur containing amino acids. Also, the product selectivities were affected by the sulfur bearing amino acids. We also used DFT and MD simulations to understand the energetics and mechanism of amino acid and protein interaction with the metal surfaces. Finally, I would like to acknowledge all the funding agencies and the HPC facility at IIT Delhi for facilitating my research.